All right, so everyone, hi. Welcome. Thank you so much for being patient with our delay. There's a lot of things to set up, and I don't know if you guys can see, but I have my Live in Farms lab coat today, and we're going to run through part 2 of the microalgae webinar lesson, and I'm going to be taking you through a few of the experiments that we've done with the students, and hopefully you enjoy yourself. It's going to be a little bit hectic today, so please bear with me. Okay, just as a reminder, my name is Deidre, and I'm the webinar designer and the webinar instructor here at Live in Farms. Today, we're going to be doing, like I said, lesson 8, which is output poop to microalgae. And this is going to be part of our ongoing Your Waste to Edible Greens webinar series. Okay, so just as a recap, if you are joining us for the first time, during the last webinar, we talked about microalgae. We talked about the link of microalgae to the Hive Explorer. Why are we growing microalgae? We talked about the benefits of using mealworm poop and why is it so nutritious and why is it so helpful for plants and algae to grow. And also we built a photobioreactor, which is something you can do in your own home. I'll recap the different elements of that again later on, and yeah, let's get going. Okay, so first of all, why are we growing microalgae? So the most simple reason that we're growing it is that it's easy to grow. And you can actually grow it anywhere. It doesn't matter if you're in a rural environment or an urban environment. You can grow microalgae really easily, and it doesn't take up a lot of space. And another reason, which we'll go into more depth in a bit, is more about the sustainability aspects of microalgae. And paired with the Hive Explorer, we use the microalgae lesson as a way to teach students about one of the different possibilities when it comes to sustainability, and that we hope to inspire them to go down different rabbit holes in this entire sustainability field. And microalgae is just one of those rabbit holes, because there's a lot that you can do with microalgae. And finally, one of the other big reasons is that it's one of these renewable resources. And I'll explain more about how it is a resource to us, but just know that it's alive, it's growing, it's easy to grow, and you can do it anywhere. Okay, so if we're going to talk about microalgae and sustainability, we need to also talk about air pollution. Air pollution, if you're here, you probably already know, but I'll just recap. It's a major concern when it comes to living in urban areas, actually even rural areas at this point. When we're talking about anthropogenic or human-made air pollution, there's a lot of different types of air pollution that we can talk about, and it really causes a lot of different health effects. It ruins the natural environment, and there's a lot of reasons that this is a big concern and a growing concern to people all over the world. And even when you're like me and you start to study air pollution and sustainability, we knew about the effects of air pollution on the climate for like a little less than 100 years ago. So this is not anything new. This is something that we really need to urgently address, and we're hoping that we can inspire students to really understand that this is a problem that is now, and there are natural solutions and ways that we can cultivate those natural solutions in order to help us get out of this really big mess that we're in. Okay, and when we're talking about air pollution, there's a lot of different causes. Air pollution is a very general term for what we're putting into the air that in most cases is not found naturally, or is found at higher levels than are found naturally because of the different human activities that we do. And one of the biggest ways that we tend to pollute the air has to do with the burning of fossil fuels. This is not just the combustion engine in cars, but also for creating energy, for example. where I come from in Indonesia, in Jakarta, it's one of the places where, at least at the time of this webinar, it's one of the only places where it's actually been allowed for them to build coal plants so close to a mega city or to a major city. And because of that, there's a slew of health effects that affect not just people in general, but especially the children and the elderly, who are the most sensitive to these air pollutants. Okay, so and then even just factory emissions, and actually air pollution is caused by almost every activity that we do, and we don't even think about it, right? Air pollution can happen not just because of like people driving down the street or driving down, wherever, but if we think about like the food that we eat and how far it has to travel, that also generates a lot of air pollution on the way for the food to get here. And it's not just the cars, it's also the planes, it's the boats, it's a lot of different ways that we move goods around the world will produce air pollution. So this is a big problem that it's very difficult to even fathom unless you're studying it all the time. And when you do, it's a lot. So what are the types of air pollution? We have carbon dioxide, of course, that's the most famous. We have methane, which we've talked about and we'll discuss a little bit more. Again, we have sulfates. If we think about air pollution, and we tend to imagine kind of like that brown layer that is just kind of sitting in the horizon, that's generally because of the sulfates in the air. And then we have the nitrous oxides, that's NOx, right? These are really, when they will start to react with each other and create a whole new level of pollutants. And so those are also generated by a lot of cars and those kinds of emissions. And then we have particulate matter, or PM 2.5 usually, or PM 10, PM meaning particulate matter. All that is those things in the air Hello? Are you guys with me? Okay, apologies. A small technical difficulty. Okay, for now, what I'm going to do is camera so we can keep going. All right, I'm back. I'm so sorry about that. I completely forgot to plug in my computer, but you know what? We're going to keep going. Okay, so where was I? I was talking about the different types of air pollutants, right? We've got particulate matter, we've got CO2, we've got methane, we've got nitrous oxides, and we've got sulfur dioxides. And for particulate matter, which I think I was talking about before I cut out, particulate matter can come in 2.5 or 10 micrometers, but it's just anything that is in the air, floating in the air, that is of that size, and that's generally what's going to get caught in people's lungs and cause breathing issues, asthma, allergies, things like that. Okay, so all right, so for another reason why air pollution is quite bad, not just for our health, which is obviously a really big issue, it's also for the fact that it accelerates and leads to global climate change. As many of you probably know, that there's this greenhouse gas effect, which actually causes basically you have the atmosphere, and the ability for the atmosphere to take in solar radiation as it comes towards the Earth, and then it bounces off the surface of the Earth and gets caught in the atmosphere, at least some of it, and some of it gets released back into space. The atmosphere serves a very important role, which is to keep the Earth warm. And it actually, because of it, there's some good parts of it, right? It helps us, it helps the Earth allow for life. Otherwise, it would be very, very cold, and that's what you see in a lot of different planets that you would think would be habitable, but because they don't have an atmosphere, they aren't able to stay warm, and they aren't able to support the temperatures necessary and the humidity necessary to foster life. So the greenhouse gas effect becomes a problem when there's all this extra air pollution and air emissions that go in and adds to the stratosphere and the atmosphere's general ability to maintain heat. And it increases that effect so much so it ends up being too hot for us. And that's why global warming was the original term used. Although, as you probably know, there's been a lot of back and forth. There are people in colder areas that say, oh, it's getting colder here and it's not getting warmer, so there must be something wrong with this concept. No, overall, the Earth as a whole is getting warmer still, but we do tend to use the term global climate change to eliminate some of that confusion. Okay, and the greenhouse gas effect gets worse, of course, with carbon dioxide, which is a really important gas to consider. But another really important one, which we have, again, talked about before, is methane, which has up to 40 to 100 times the warming potential of carbon dioxide. And so you don't need as much methane in the air, and actually methane naturally doesn't occur in such high quantities in the atmosphere. But the problem is that we are also generating methane along with all those other pollutants as part of our regular processes that we have, and that makes the atmosphere even more warming than would happen if we only did CO2, right? And we talked about food waste you can check out webinar number 4 and number 5. Those talk about food waste and processing your own food waste so that you avoid putting that food waste into the landfill, where it'll start to release methane as it's breaking down. So not good, right? And there's different ways that we can plan to reduce all these air pollutants. The food waste processing is one way, removing the food waste from your waste source. And also, big ones would be reducing emissions. That's actually probably the biggest one, to be honest, is just not using as many private cars, investing more in public transportation, things like that, moving away from burning fossil fuels, of course. And the one that's important to us today would be to capture carbon in the air, right? If we can remove some of that carbon, and yes, in methane, it's CH4, there is carbon in there too, so that would count. And removing the carbon dioxide, then we can get rid of some of those greenhouse gases that are in the air. Let me turn my camera back on. All right, so you can see my face again. And let's move on. So a really great benefit of the microalgae and teaching students about the microalgae is its potential as an air purification method. And microalgae are tiny, well, micro, right, means that it's small, it's microscopic. These are tiny single-celled organisms, and they tend to live in water, so this can actually be fresh or salt water or even actually wastewater. They survive there too. And they're quite powerful as air purifiers. And this ability means that we can harness this potential to help solve some of the really, like I said, the big problems that we're facing. And it's a really promising, it's promising in terms of thinking of a sustainable future, not just because of, oh, yes? Oh, yeah, go ahead. So it's powerful not just because of its ability to help us with this air purification problem, but also because of the potential with renewable biofuels, with medicine, with cosmetics, with fertilizers, animal feed, and even human feed. And so let's look at this absorption ability and why it's such a great potential for us. Okay, so when it comes to carbon dioxide and it comes to these other pollutants, the main thing that microalgae run on, and we can think of this in terms of a system, again, is that it runs on, the process that it runs on is photosynthesis, which is the same as that of plants, right? Photosynthesis, just as a reminder, takes in carbon dioxide, it takes in water, and it also runs on the solar energy from the sun, and it turns that into sugar and oxygen. And in doing so, it actually also removes the air pollutants. So even though the air pollutants are not part of that photosynthetic equation, like CO2 is, but none of the other ones are, it does also purify the air from those other types of pollutants that we talked about. Right? And the benefit is that it produces O2, which is great for us, right? And if we think about how long Earth was able to support life, that was about the Earth, well, life was able to be supported on Earth for about 4.5 billion years, which actually, when you look at the lifespan of the Earth, which is about 5.56 billion years, that's a really long time for Earth, for life to be around. That's on geological time, that's actually quite fast. And microalgae were one of the first to be able to live in the water. And because of this process of releasing oxygen into the air and consuming the CO2, it actually helped plants and animals and us eventually, like our ancestors, to be able to colonize the land. And this, several million years after they evolved, they led to the oxygen revolution. And so the atmosphere that is able to support our breathing and our living, 70 to 80% of that was created by aquatic plants, and most of which were algae and microalgae. So really, we owe our ability to live here on land, nice and dry, to these really simple and really tiny and powerful microorganisms. Okay, and so what about when it comes to farming, right? Farming, we've talked about farming or I guess in general, the more problematic parts of large-scale farming and the food system. And so how does microalgae kind of play into that? Well, first of all, you can grow microalgae in a lot of different types of water. Like I said, it can be salty, it can be fresh, it can even be wastewater. And it also doesn't require soil. So it means that it's not competing for the land where we would normally be growing crops or livestock. So that's a great part of it. And it's also just, it's space efficient in general. So you don't have to do it out on a big field. You don't have to do it on even a fertile land. You can see that there's even projects going on in California and in the Middle East as well, where they're growing these huge entire vats of microalgae out in the desert, because as long as you can get the water there, you don't need the soil. Okay, and if we think about how much land or fertile land that our food is taking, most of it goes actually not to our crops, but to the livestock that we raise, because we don't only need to have space for the livestock, we also need to have space for the crops in order to feed our livestock. So it takes up a ton of space, but at least we don't have to compete with the microalgae for this. Okay, another benefit is that it's very fast growing. Microalgae starts to go through cell division at about every 12 to 16 hours, so like every half a day. And when you first grow a colony of microalgae, you'll find that the population will double within the first 1 to 2 days. And so they grow very, very fast, and you don't need a whole lot to start off with, actually. Another benefit is that when you grow the microalgae, you're going to get, of course, the microalgae colonies. So even though they're single-celled organisms, they do like to grow in multitudes, and they will clump together in this big colony. Even when you look at seaweed, seaweed is actually an example of algae. It's just algae that likes to live in colonies, which is why you're able to see them. It's just millions and millions of these very simple creatures, organisms living together, so that you can see something that is actually visible with the naked eye, as opposed to being just microscopic. And algae biomass in particular, you can actually sift out the algae, and that can be used to create biofuels. That's another part of this, that it's predicted by the US that I believe it was the US EPA to say that biofuels and microalgae biomass can replace almost any engine that's currently running on diesel fuel. And there's a lot of issues with diesel, partly because it competes with biodiesel, because it competes with the farmland, because biodiesel comes from usually corn. And so suddenly you're not just growing crops, you're not just growing crops for the animals, and you're not just growing animals, now you're adding biodiesel to it. It's a lot, and we don't have a lot of space for that. So microalgae is a great alternative if you can also run it on those same engines, because then you don't have to go through and change every single car that's coming along. Okay, and just a fun fact, as I've mentioned, microalgae really runs on photosynthesis. And one of the benefits is that it's actually more efficient than trees in converting carbon dioxide and water into sugar and oxygen. And its ability to catch CO2 in the air is about 150 times more efficient, or has a higher ability to do so than trees. So that's pretty cool, I think. You really don't need as much space to grow them. They grow much faster. Another fact is that microalgae can absorb all types of pollutants. I've kind of briefly mentioned this one already, but it's not just carbon dioxide. It's also taking in the carbon monoxide. It's taking in the nitrates, the particulate matter, and also the volatile organic compounds, VOCs. Those are quite dangerous, just like the nitrates, because VOCs are actually the types of fumes that you get when someone paints, for example. That kind of like smell, that's a type of VOC that comes out, and just from its name, you can tell it's actually quite volatile. And so it'll react with the other chemicals in the air, and we get things like ozone and other types of pollutants that cause really bad smog. That's a huge problem in Hong Kong, but it is in a lot of other places as well. So you can start to lower the emissions of like cars, for example, but if you don't get rid of some of these other pollutants in the air, then you're gonna still wind up with pollutants reacting with each other. And so stopping emissions is not the only, it's not the only thing that we have to consider, unfortunately. But also when it comes to these air pollutants, I think personally I feel like one of the great things is that it turns this big problem of CO2 and other pollutants, and we make it part of the solution, right? So we can think of these as really big problems to solve, but actually Mother Nature already gives us a lot of different ways to utilize things that are wasteful and to turn them into an opportunity as opposed to a detriment. And that brings us to this third fun fact, that microalgae is really not that wasteful for cultivating it properly. It runs on a lot of different wasteful products, such as wastewater. Like I said, you don't need a particularly clean water source to do it. And it also runs on animal castings, so animal poop or mealworm frass, which is what we used. And it also uses the air pollutants. So microalgae are really, really helping us clean through a lot of things. And they can even be used to filter your office environment, for example. You can run a very small photobioreactor there with no problem. All right, so when it comes to the Hive Explorer, the great thing when we're talking about sustainability aspects is that you can pair it with the Hive Explorer and use the outputs from the Hive Explorer as an input for the photobioreactor. And you can use recycled plastic bottles. You can use the natural light coming from the window, the solar energy, to run your photobioreactor. And you can compost the dead algae that will eventually start to form anyway, and you can put that into the compost and eventually have it feed your hive once you process it properly. So this is kind of us finally coming to close the loop on some of the systems that we've been talking about. We've been talking about how the hive is linear, and you need to add to it and help it so that it can become a circular process. And this is one of the ways that you can help to do that, plus some of the other tools that we gave you with the other webinars. Okay, and I think one of the best things is that you can really grow it anywhere, like I said. So what I'm going to share with you now is, actually, I'm going to give you like a sneak peek. I obviously can't give you guys the full worksheets yet, but we have run this workshop with several schools, and one of the activities that we do is we show CO2 absorption. And so what we do is that we have two photobioreactors, one with water, one with frass and microalgae growing in it. And we turn on these CO2 meters to see what the readings are for those. And we see over time, I mean, I actually have it set up. This part of the reason why it was so late last time, earlier today. I have this set up here. I didn't turn the meters on yet. But you can see, let me just stop this for a second. Okay, so you can see here that we have these two setups. We have water, right? We have the microalgae. There's poop in here as well. It's pretty murky, but I set this up during the last webinar, and it's going pretty well. So let me turn on the meters. And I'm not going to be able to show you exactly how it runs yet, but we can definitely let it sit for a little bit. Oops, I need to take this out. Okay, we can let them sit. Okay, and just in case you didn't join us last time, the photobioreactor is actually just this piece right here, plus this air filter. We've got an air filter, a hose, and then it connects to an air stone, which you can see here. and it's just putting bubbles in here. And then we added a mixture of frass and water and a tiny bit of concentrated microalgae. And that is from a solution that we had at the office, but you can actually go to a river or a stream or even the ocean inland, and or not inland, but like closer to the shore, and just take a small sample of water and pour that right in. And eventually it should start the colony by itself. Something to note, I actually have another bottle here as well that I also prepared. This one was just with the, oops. This is just with the microalgae and water with no frass. You can see these two side by side, and you might think that this one did a little better than this one just because it's more green. But actually, when I was looking at it earlier today, if you look at the bottom here, the microalgae tends to start growing right at the bottom, and there's not a whole lot down there. It's already green because I put in a starting culture. But actually, if we look at this one, I'm gonna look down, there's actually a lot of different green bits in there. I'm not sure if you can really see, but I can definitely send more photos if you're interested. This is a really great activity we tend to do with the students, and they love the hands-on experience. I'm just going to set those up so we can let it run for a little bit. Okay, and all of these materials, you can use a bottle that you just recycle. These are just bottles we had around the office. And actually, the rest of these materials, excluding the CO2 meters, can be bought at an aquarium store or a pet store, or just like the market if you happen to live in a place that does that. Okay, you're going to see my face again. And so, like I said, we have some worksheets. And what I have shared with you is actually we tend to run these activities. Well, we have the students help us make this setup as part of a demonstration. We have some students help us out. And then we run it in the background while we teach the rest of the lesson. And as you can see on the slide, that we check on it and every 10 minutes for about an hour. And then we have the students plot and graph the progress of the carbon dioxide levels in these two containers. You can see here, this is actually one of the schools that we worked with, and you can see the two of the students helping us out. And there's just another photo of the setup with the meters. Okay, and so this is the next big activity that we generally have. Sorry, I'm running out of time a little bit, so I'll hurry it up. Basically, this is going to be the microscopy activity. I took a culture of the microalgae colony that we have set up from the photobioreactor. And during the time that we had them in school, we happened to be in schools that had setups where they could use the microscope to attach to the projector, which unfortunately we don't have. But I have prepared some slides for you, and I've prepared some photos. And I will also really quickly show you how you can set up your own microscope at home. We tend to think of these things as pretty fancy lab equipment. But I will tell you, as someone who's had to work in the field and has had to be really creative with how I do things, that these guys are pretty sturdy. Obviously, you need to be careful of you need to be mindful of the lenses that we have. But in general, you can buy these secondhand as well. And so while they tend to be quite expensive new, they last a really long time. And you like I said, you can get them secondhand just like clothes, and actually they're probably more sanitary, to be honest. Okay, so first let me get you acquainted with the microscope. Okay. During this time, I do also want to encourage you to send in your questions. We do have moderators here who can help flag them so that I make sure to answer them. Okay, so please go ahead with those while I talk about this and we can loop back to them later. All right, so here's your microscope. This is a type of light microscope because it uses the light, which I'll have a light source later, and it funnels it up to the top, or it reflects it up to the top, and you get to see the different magnifications here, right? That's something we teach the students. I don't have to go into too much detail, but the red, it goes red, yellow, and then blue being the highest magnification. And you've got these two different focal points. We've got this is the primary focus knob, which will move the lens up and down. Okay, and then this one right here is the fine focus. Generally, that's what you do just to get the image completely crystal, especially when you're using the higher magnification. Okay, let's see. So I've got my lenses here. Okay, when you get a microscope, you tend to have a lot of different parts. This is just an extra 2 times magnification in order to determine the magnification that you're looking at. I mean, we don't require it of the students since they're quite new to using these, but in general, you would determine the magnification. You would look at your specimen under the microscope, you would write it down. A lot of times you would either take a picture or you would draw it. I like to draw it. When you're out in the field, it's better to just draw. Then you can get everything down at once. You determine the magnification from the top right here. There'll be a sign that says here, it says 10 times. Then you multiply it by the magnification here, which is 2 times, so that's 20, right? And then you get the magnification from one of these. Let's see, this one is times 4, so 4 times 20 is 80, right? And so you would know that the magnification of whatever you're looking at is 80. You'd want to write that down so that you know how zoomed in you are. When you're preparing a slide, you're going to need a big slide, the glass slide. This is actually one we'll use later. Okay, got a glass slide right here. We've got, took a little bit of the specimen right there. This is from the frass and the algae. And we have these cover slides as well. Okay, so first thing, I'm going to take the pipette. You want to squeeze it first, right? And you're going to just only take a tiny bit, and you only actually need 1 drop. There you go. That's perfect. Okay, and now I'm going to take a cover slide. Cover slides are made out of glass. When we do this part, we tend to do it ourselves as opposed to having the students do it because it is very, very fragile. They're kept in these boxes, and when you hold them, generally you use tweezers. If you're ever out in the field though, like I am a lot, then you need to hold them from the sides. You don't want to touch the main part of it, right? You don't want to make your slide dirty, and you have to touch the edge, and then you slowly drop it, and the water should spread all the way through. See that? Okay. That's good. And usually you'll want to suck up any of that extra water because you want the slide to lay flat. So I just have a tiny piece of paper. You can use a piece of tissue as well. And I'm just going to put it right at the corner. You can see it's just going all the way up now. Right? Okay. And that should help your slide lay more flat. Cool. When I've had to do this in the field, literally we'll be sitting on the floor without our shoes. We'll be like having to wash our hands right out the back, but I've done this in a lot of rural communities where unfortunately clean water is not readily available all the time. And I mean, but science still happens out there. You need to do science out there, actually. Okay, and you can see this is actually very simply our slide. Most of the time, you're actually going to want a dye in there. You can, for a natural alternative, because you can use different like synthetic dyes, you can use beets, you know, because beets have this like red pigmentation. You can use it to dye your specimens. In this case, you don't need to, right? Because they already have this green pigmentation. So we should be able to see it under the microscope already. Okay, let's see if I can find it. So it's gonna be a little tricky. You guys are gonna watch me while I do this, but I will share the final photo with you in a second. Okay, and while I have that up and while I'm looking for it, you can take a look at the photos that we've taken from some of our other experiences with schools. And I think something that's important to note is, like I said, microalgae is a single-celled organism, even though they do tend to live together clumped up. And when you see like the middle bottom slide, middle bottom photo, I mean, all of these are microalgae, but the middle bottom photo shows what it's like when they are separate. And you'll notice that they actually look quite similar to red blood cells, like hemoglobin. And they're just single-celled organisms. So it means they're entirely functioning with only one cell, which I think is pretty cool. All right. Let's see if I can do this. I need my light. Okay, this is another thing is that we need a light source. And what I'm using is actually a light that we will be using. This is just something we had at the office. It's like a long LED light. You don't have to use this. It depends on your microscope setup. Sometimes the light is included as part of the microscope, but not for everyone. And it's not a big deal if you don't have like a built-in light. You can actually just do it with it. Just find what you have at home, because honestly, that's one of the more sustainable things you can do anyway. Okay, and what I'm doing is I'm just taping it down because our light source is so big. I don't want it to move around too much. Again, we have to be quite creative when we're out in the field. We can't just go out and buy a lot of things. We have to bring everything we feel like we need into the forest or into these rural areas and hope that we have everything. And if not, we just gotta figure it out. So, all right, now you can see the setup. Got that light at the bottom. It's pretty bright, but it's just going across and it's taped on both sides so it doesn't move. Okay, and you need a lot of light when you look at your specimens because because of the magnification, it makes it harder for light to come through. Oh, I already found something. Wow. Which means that as you go up in magnification, the hole in your lens is going to get smaller and smaller, and that means it's less and less able to let light through. And you need to help more and more light to get through. All right. Yep, I got something. So I'm gonna take a photo. Oh my gosh. Then take a photo, and I'm gonna show you guys. Okay. All right, so this is the photo I got. Our colony is still quite new. Oh, so it's not quite green, but you can see that there's these different blotches. That's actually the clumped up microalgae. And you can see it's actually quite similar to the other pictures that we got there. You might notice that there's that blob right in the top middle, and that is just the microalgae kind of clumped together. And so it's quite difficult sometimes to separate them out, and we try to suspend them in water so that they can spread out a little. Okay, I hope that was interesting. I know it was a little, I guess the term is MacGyvered or something like that. I'm not quite sure, but anyway, that's how I'm used to doing this kind of field work. And I personally love using the microscope, and we really try to instill that with the students as well. We find that the microscopy activity is one of the most engaging for the students. And when we do have the ability to take the microscopic images and put it on screen, like that setup that we have in the presentation, we find that the students are super engaged and they love seeing what the world is like under the microscope. And we're happy that we can share that with them, especially with the power of mealworm frass and the Hive Explorer. Okay, so that's it. Thank you for listening. Okay, let's see the questions actually. Okay, so Schwan asked, how do we seed our microalgae to get started? Sorry, I missed the last webinar. No problems. So as I mentioned, so you can actually take a small water sample from a nearby stream, or if you don't even have that, that's okay. You can, so here, I have an example. So what we do is that even though we did have a microalgae culture already, to help it further along, for the first 2 days that we had this setup, we actually removed this hydroponic foam so that there was a greater opening, and then we actually let this run for the first few days without the foam in place. We put it by the window. So what we're doing from the offices is actually keeping the window open when we do these, and it allows any of the microscopic organisms or microscopic algae in particular to come in through the window and into the bottle. And you don't have to worry too much about other things coming in as long as you make sure to close the window at night and you put the foam thing back on once it's done. We're making this a great habitat for these guys. And if anything ends up in there that you don't want, which actually has never happened for all the different times that I've done this, then you can also just filter them out. So yeah, I hope that answered your question. Let's see if anyone has any more. All right. Yep, I answered that. And yeah, I can send more photos later. Yes, please check out our discount code. Since you are joining us, you get an exclusive discount that you can't get anywhere else. So it's just a thank you from us for joining us. We would also really appreciate your feedback. And yep, so you could fill out the webinar, especially if if you've actually attended a few of these webinars. That's super helpful for us to know what we're doing right, what we're doing wrong, and how we can improve. Okay, and final thing before I go, I have been posting on the blog, on our Hive Explorer blog, and I talk about different aspects that are somewhat related to the webinars that we do. And this previous blog post was about microscopic organisms and especially microscopic insects that you can find. So please check that out as well if you're curious. And yeah, I hope to see you guys next time. Thanks so much. I'll leave this up. I'll leave the slides up and you can check that out. So till next time, bye.